Been a lot of consternation, I guess, if that's the right word, with regards to identifying your unknowns. And you guys have a great resource available to you. It's called the internet. When I was in school, I didn't have the internet. You know, I didn't even have a typewriter. We didn't have paper. I had a stone and some chisel. You know. Anyway, I got online and I found a site here that you might find useful. It provides flowcharts for helping you identify your unknown. So the first page just kind of gives a description of the different groups, but then you go to the following pages, and here's a flowchart on gram-positives and gram-negative cells. Then further down we have a flowchart on gram-positive rods, and then you can follow this to help you identify your microbe. Go further down, there's a flowchart on the bacillus strains, flowchart on gram-positive cocci. So I would encourage you to look up this website here. Let me go back. All I did was type in Bergey's Manual of Determinative Bacteriology. And you can see there's lots of different pages that you can go that might help you with the identification of your unknown. You can use those flowcharts to help you get narrowed down to one or two particular microbes. And then from there, maybe you type in the microbes of interest and see what kind of description comes up for those microbes. So just something I wanted to point out to you that's available. So this is a little video on the new testing for TB.
Tuberculosis is a global health problem. It is one of the most important preventable causes of death and morbidity in the world today. In the United States, the disease continues to be a significant challenge due to immunosuppressive conditions, immigration, and non-compliance with testing and treatment protocols. Mycobacterium tuberculosis, MTB, is a slow-growing intracellular organism, which makes culturing the organism slow and unreliable. Latent tuberculosis infection, LTBI, occurs when people carry Mycobacterium bacilli in their body, but the organisms are dormant and still in small numbers. These mycobacteria are controlled by the body's immune system and are still in small numbers. In this state, the MTB do not cause disease or any TB symptoms. Individuals with LTBI are not infectious. They would be negative to most TB tests, including culture and smear microscopy. Small nodules are occasionally seen on chest X-ray. Until recently, the only test capable of identifying LTBI has been the tuberculin skin test, TST. The most commonly used TST is the MAN2 test. This involves an intradermal injection of purified protein derivative, PPD, into the arm. PPD is a mixture of proteins derived from dead M. tuberculosis cultures. An immune reaction occurs within 48 to 72 hours, at which time the induration is measured. This test requires 2 patient visits. In addition, the test requires up to 72 hours to get the result, and false positives can occur in BCG vaccinated subjects or following infection with environmental mycobacteria. The DNA of Mycobacterium tuberculosis has a region of difference called RD1 that is not found in BCG and most environmental mycobacterium. The RD1 area of MTB encodes for 9 proteins, including ESAT6 and CFP10. These two proteins are used in the T-spot TB test to challenge T cells. If the T cells recognize these proteins, they secrete interferon gamma. The T-spot TB test is a simplified version of the enzyme-linked immunospot ELISpot assay technique. It identifies M tuberculosis sensitized effector T cells activated by the presence of CFP10 and ESAT6 antigens from the RD1 region of MTB. Effector T cells are quite short-lived. Their continuing presence indicates the cellular immune response is currently encountering and fighting a pathogen somewhere in the body. Measuring the presence of effector T cells in the sample therefore diagnoses an ongoing infection. In contrast, the presence of memory T cells would only indicate that someone had been infected with a pathogen at some point in the past. Blood from the patient is centrifuged, and the number of peripheral blood mononuclear cells, PBMCs, are counted. The sample is diluted so that a standard number of PBMCs are added to each well. PBMCs are added to a pre-coated well that contains antibody to interferon gamma. Cells are incubated for 16 to 20 hours. In the presence of activated effector T cells, the TB-specific antigens, ESAT6 and CFP10, stimulate the release of interferon gamma, which binds to antibodies to interferon gamma on the base of the well. Wells are washed, and a secondary antibody to interferon gamma is added. Following another incubation and washing step, the substrate is added. This produces spots on the well floor where the interferon gamma is secreted by T cells. These spots are enumerated to provide the test result. The T-spot methodology provides an effective means of identifying TB infection. The test is highly sensitive and reliable in targeted testing groups. T-spot TB provides comprehensive testing coverage for all high-risk groups, including healthcare workers, immunosuppressed individuals, TB contacts, and TB suspects. The assay detects interferon gamma produced by T cells in response to RD1 specific peptides, so the test is also very specific. T-spot TB test results are not associated with immunocompromised status. This is because the T-spot TB test requires a standard number of PBMCs to be used for every patient, thereby minimizing the effects of low cell counts. The T-spot TB test is highly specific, as it does not cross-react with the BCG vaccine and most common non-tuberculosis mycobacteria. Benefits of the T-spot TB test are its efficiency, requires one single blood sample, it is fast, next day results, and it is one and done, no second visit required to read test results. Stop TB. Start with T-spot TB.
Comments, any questions? So this is a new technique that they'll probably start deploying, I would imagine, pretty soon across the board, simply because it seems like it's more specific. We'll be able, if you think about the implications here. Right now, we don't vaccinate for TB in the United States simply because it becomes difficult to track who's been vaccinated and who's actually been infected. Because of the specificity of this, so that now that it only targets people that are actively fighting an infection, meaning that they have effector T cells that are active for that infection, we could start vaccinating for TB we wouldn't have to worry about the false positives that you might get from somebody that's been vaccinated, because we're not looking for that particular antibody anymore. We're actually looking for active effector T cells. And so it's a much more effective method for looking for TB. Whether or not we will start vaccinating for tuberculosis, I'm not sure, but you can understand why this would be a preferred method. Any questions, any comments? Okay, so today we're talking about skin infections. If you think about it now, your skin is coming in contact with the outside world. So anything that's in your environment, chances are your skin is going to encounter. If your skin is intact, then you're pretty resistant to infection. Most microbes are not going to be able to penetrate past the skin unless there's been damage done to the skin. We will see that there are some microbes that can infect hair follicles. We'll talk about those microbes. And then in some cases, the infection can be inhaled, make its way into the blood, and then move into the skin. So you can have skin infections that are carried there by the blood. When we think about the anatomy of the skin, most of you are probably familiar with it. We can talk about it coming in three layers: the epidermis, the dermis, and the subcutaneous layers. Now the dermis is going to be the outermost layer, and it's composed of keratinized cells. They're dead cells. They slough off constantly, and in sloughing off, they take microbes with them. Below that, we have our dermis, and inside the dermis, you're going to find where the hair follicles are existing. I'm going to go ahead and draw one here. So this is the epidermis, and we've got several flat layers of epithelial cells. And then down here we have the dermis. And this is where we're going to find the hair follicles. Attached to this, you're going to find glands, sebum glands. In addition to that, there'll be sweat glands that are located in the area. And then down here, below the dermis is the subcutaneous layer, and this is where we're going to find a whole bunch of fatty deposits. So that's pretty much the anatomy of the skin. Again, its function is really to help control temperature, loss of fluid, protects us against our environment. We find numerous sensory receptors there for temperature, for touch. When we consider our normal microbiota, those microbes that we find on the skin, generally in everybody in the population, of course this will vary depending on the individual, and climate can have an effect on your normal microbiota as well. But there are three main inhabitants: diphtheroids. So we've encountered these before, and we'll talk about a specific one today. Staphylococci, or Staphylococcus are found there, and we'll find some yeast cells as well inhabiting the skin. For the most part, as we keep saying, our normal microbiota, these guys are going to be harmless. They're not going to cause any illness. So generally, but on occasion, they can be opportunistic. So if you have a depressed immune system, they can make you sick. Let's consider the diphtheroids first. They resemble Corynebacterium diphtheria. They're gram-positive microbes. They generally have a low virulence, and they do not produce an exotoxin. There is one microbe specifically we want to talk about. Generally, these microbes found on the skin break down your sweat. One of them in particular is, I'm going to spell this wrong here. Propionibacterium acnes. Everybody has this microbe on their skin. And it is a diphtheroid. Okay, it is a diphtheroid. This microbe here, because of the name, you might think that it's responsible for acne. It's not really responsible for acne, but it is associated with breakouts. So the microbe itself grows within the hair follicle. So Propionibacterium is one of those